Hi class, today we're going to be looking at DNA replication. This is chapter 10, section 3 in your book. And what we're going to look at is how does the DNA replicate? Now we've talked about in the past why it replicates, and the purpose is for mitosis, to get an extra copy of the DNA so that when it goes through cell division, we have that extra DNA in order to have a new cell eventually. So the process of making new DNA happens in that S phase of interphase. Here we see occurs when chromosomes duplicate before mitosis and meiosis. And it will be in the S phase of interphase, where it's that synthesis phase that makes the DNA. It will make an exact copy of the DNA. Now, every now and then there could be some mistakes, and we'll talk about how that can happen. But overall, it is an exact copy of the DNA that's already there. Hydrogen bonds between the bases will break, and the enzymes will basically unzip the molecule. And you can kind of see that here. What it's representing in this picture is that we have this kind of breaking point between the two strands of DNA, and then we have new strands of DNA that are being formed along the one-sided ladder, I guess you'd say. So we're making the second side of the ladder. And so we'll go through the process of what enzymes do this as far as kind of break this apart, and then what enzymes bring in material that will make the other side of the DNA. This process occurs all over the DNA strand at the same time, not just from one end to the other. So it doesn't work quite like a zipper on your jacket where you go from one end, from the bottom of the zipper to the top of the zipper. Instead, it will be happening all throughout the DNA at the same time in little sections. It makes the job quicker and a little more efficient. If you're in your notes packet, that information just went under the DNA replication set. If you skip then to the next page on your notes packet, we're going to go through the steps. Now these steps are on page 201. If you'd like a visual, a little bit more visual, or to read about it, you can go there. Enzymes called helicases separate the DNA, so this is going to be step 1. The enzyme here, helicase, you will need to recognize that word again. You're going to see it, and you're going to need to be able to describe what it does eventually. So they separate the DNA strand, breaking the hydrogen bonds. This H stands for hydrogen bonds between the A's, T's, C's, and G's. And that position, or that spot, is called the replication fork. So this is helicase here, which you can see it's labeled. And then that is creating what is called the replication fork. At step 2, enzymes called DNA polymerase, so you do need to know that as well, they add the complementary nucleotides. So you have this DNA polymerase, I'll just put DP, that comes in and starts adding the new nucleotides, the ones that would go across from this strand, this original strand. Step 3, DNA polymerase falls off when it's done replicating, and the result is an identical strand of DNA. Now, in case there's mistakes, we have the enzyme ligase. It will kind of follow in behind, and it fixes any mistakes that happen to be there. Now there is a little more detail to this. One side of the DNA will get copied from end to end, so from start point all the way down. The other side is going to be done in fragments. So it'll be done in a section, and then it'll be done in another section, and it'll be done in another section. And then there's these gaps, and these gaps will get filled in by ligase. So ligase comes in and fills in this space with the correct nucleotide sequence. These fragments that are on one side, that are made on one side, are called Okazaki fragments. So if you happen to see that term, that's what that means. DNA polymerase came in, one side went from end to end, the other side did these fragments, and those are called Okazaki fragments. You can see here in our picture kind of what's happening. So we have helicase right here. That's kind of our important enzyme to make the replication fork. It's making a replication fork right here. Right here where it's breaking that apart. DNA polymerase is coming in and adding the new nucleotides. So you can see here, it's adding new nucleotides. These are nucleotides that are being brought in. And then we have these fragments being put together on the opposite end. DNA polymerase is still doing that, but it's Okazaki fragments, and then ligase will come in and add in the empty spots. As well, ligase will come in and fix any mistakes that might happen through DNA polymerase. This process you definitely need to know. You need to be able to explain it as well. So make sure that you go back through and really understand it as something that you can communicate back to me or to someone else. Now you have this picture in your notes packet. It didn't copy very well, so you may want to fill in, pause here, and then fill in what you can't read on your notes packet. One last little part about this is it's called the semi-conservative replication, meaning that we have a new strand and an old strand in our set of DNA. So currently, if we look at this picture, this is our parent strands, the original DNA, and then they split at this point called the replication fork, and this part here is the old strand, this part here is the old strand, and then we have this complementary new strand that's being added in, and a complementary new strand that's being added in here. Those then, when we end our DNA, when we get done with our DNA, this is called semi-conservative in that we have an old strand and a new strand on each sets of our DNA. Show you this replication idea again in a different view, if that helps. So the parental DNA molecule contains old strands, hydrogen bonded by complementary base pairing. Region of replication is where that DNA polymerase will work. Parental DNA is unwound and unzipped. New nucleotides are pairing with those in the old strands. And then replication is complete when each double helix is composed of an old strand and a new daughter strand. Okay, that's the end of this section on DNA replication.